The central question in developmental biology is how you get from an egg to an adult organism. Now, if you stop and think for a moment about the huge variety of organisms that exist, you'll realize there's a problem. For example, most eggs or early embryos, such as this seed, are much, much smaller than the adult. So somewhere along the line, development must involve growth. And generally speaking, growth means cell division. You start with a single-celled egg, and you end up with an organism with millions and millions of cells. Now, the adult organism isn't just a ball of identical cells. There's lots of different cell types. In my hand, there's blood cells, skin, cartilage, bone, and so on. Or in a leaf, there are cells which allow gases in and out, cells which transport chemicals inside the leaf, lots of different types of cells. And all of these have got to occur during development by a process known as cell differentiation. Now, the third difference between the egg and the adult is very obvious. Look at any adult organism. The shape is a lot more complicated than a simple oval egg. So this generation of shape, so-called morphogenesis, must also be a component process of development. So there are three component processes: growth, cell differentiation, and morphogenesis. Now, if for a minute we come down to a little more detail, take another look at the leaf. Really, I think that everything involves behavior of cells. Cells increase in number during growth, they change in type during cell differentiation, and they take up different shapes and positions to give morphogenesis. So arguably, we can consider development in terms of what happens to individual cells. Well, so much for the so-called philosophy. None of it would be worth anything unless we had some sort of experimental techniques to test our questions, and of course, test our supposed answers. So what I'd like to do now is briefly consider some of the general experimental approaches that are used. The first approach, and arguably the most important, is just to watch what happens, to use your eyes. After fertilization, the most obvious event is cell division. This period, called cleavage, leads up to the formation of a ball of cells, the blastula. As yet, there has been no increase in the size of the embryo as a whole, so cell division can occur without growth. The next phase of development is gastrulation. Here, cells from the outside move to the inside, vanishing through a plug hole, the blastopore, and inside cells are forming the three layers typical of the gastrula. So in gastrulation, the embryo starts to take shape, a process that becomes more obvious in the next stage, neurulation. Cells migrate to form the primitive nerve cord. One of the features of morphogenesis during gastrulation and neurulation appears to be the movement of cells. And of course, with further development, the outline of the embryo becomes more familiar. By now it looks like a tadpole. It's amphibian development that we've been observing. So simple observations using time-lapse film can tell us a great deal, but there are limits. If we look at an early stage, the blastula, before morphogenesis is obvious, is there any way in which we can tell what particular cells are going to become? Is there a pattern in the blastula? What we can do, in fact, is to add some harmless dye to the surface of the blastula and watch what happens. For example, if we apply a little dye to the surface of the blastula near the animal pole and allow that dye to sink in to stain some of the cells underneath, we can now allow the blastula to develop. And if we take a look at the neurula and take a transverse section, we'll see that the dye is concentrated in one particular region, a structure known as the neural tube, an early component of the nervous system, which means that this neural tube must have derived from some of the cells we originally stained. So in a sense, we can map the neural tube back to that region of the blastula where we originally applied the dye. We could, of course, apply dye to a different region of the blastula. And in this instance, if we allow this blastula to develop, we'll see that the dye is concentrated entirely in mesoderm, which must mean mesoderm can be mapped back to that particular region of the blastula. So that region of the blastula can be assumed to be presumptive mesoderm. By applying dye to different regions of the blastula, we can determine what the prospective fates of each of those regions will be. We can look at the structures in later embryos and map back those structures to the original blastula surface. And in this way, we build up a complete map of what the fates of those regions will be, a fate map. By now, we've built up a good description of development, but really know very little about mechanism. What we need are other approaches. Dr. Dennis Somerville, National Institute for Medical Research.
Here we have an ordinary hen's egg that's been incubated for about 4 days. At the same time, this other egg under the microscope was put into the incubator, and if we look down the microscope, we can see many of the features of the developing embryo. Very obviously, in the center, we have the heart, which is beating. Here, this gray pigmented area, you can see, is the developing eye. There's this balloon-shaped sac, the allantoes, which is similar to the mammalian placenta. And the bit that we're particularly interested in is this white bulbous area here, the developing limb bud. What we're going to do in this experiment is interfere with the normal development of the limb bud, and we hope that the interference will produce abnormal development that will give us some insight into how normal development is controlled. Here we have the host embryo. What I'm going to do is to cut a hole here on the anterior border, that's the head end. I'm then going to take a piece that I've already cut from the donor embryo from the posterior border or tail end, and I'm going to take this piece and graft it into there. that's the graft in place. The pin will just hold it there until the cells heal into place. I now take the egg, and because it'll go off, because we're working in these non-sterile conditions, I have to add a few drops of antibiotics, and then it can go with the other eggs, ready to go back into the incubator. So this is the normal limb. Which you can see has the hand plate at the bottom that's going to develop into the hand. And now, if I turn it over, we get to the bit that I still find exciting after years of doing this. We see whether the operation was a success. And here we have the operated limb. And you can see very clearly the hand plate with the normal hand down here. And what we have developing up here, the anterior edge, is another bulge that's going to form a mirror image hand. So this chick will have two hands, mirror image symmetry. Finally, we can look at the 10-day embryo. That's necessary because then we can stain it so that we can see the details of the skeleton and the developing feathers, even if we're careful, the muscles. And we need this to tell us exactly what is present. Have we produced a totally normal hand, or do we only have parts of the hand? This is the normal wing at 10 days. It's been fixed and stained so that we can see the skeleton, and we can see lots of other details too. You can see very clearly the elbow, the wrist region, and the hand, and humerus, radius, ulna, and this bit here that looks like a thumb is actually the second digit, and the middle finger is the third digit, and the little finger is the fourth digit. The others have been lost, the other two, just like where our little toe is being lost. You can see just there the last vestige of the fifth digit. You can also see very nicely the feather germs which are developing for the flight feathers. Now, compare the operated limb. You can see that proximally it's much the same as the control limb. We still have the humerus, the ulna, the radius, and the wrist. But in the hand region, it's reduplicated. Here we have the original host hand with the little finger, that's digit 4, you remember, the middle finger, digit 3, and the thumb, or digit 2. But we also have this reduplicated hand in mirror image symmetry. There's another digit 4, another digit 3, and in fact it shares the thumb, so it contains it in this midline position so that the entire set of 5 digits comes in nice symmetry.
What this experiment tells us is that the graft has somehow reprogrammed the cells. The limb is duplicated, not normal. So in normal development, neighbouring cells must talk to each other, instruct or program one another. If you like, I'll become a feather, you become skin. Each cell is part of another cell's environment. Another aspect of the developmental program is almost certainly genetic. A hen's egg develops into a hen. but natural or induced mutations can happen. A normal fruit fly, and a mutant in which the antennae are replaced by an extra pair of legs. We can bring about this major change by mutating one gene. Experimental manipulation of genetic inheritance or cellular environment to produce abnormalities can tell us much about what governs normal development. But here again, we can't get all the answers. Sometimes the system is just too complex, and we need to look at model systems, simple systems which model or mimic more complex ones. The cellular slime mold provides such a model. Professor John Bonner, Princeton University.
The life cycle of these slime molds is particularly interesting and, in fact, particularly unusual, and I would like to show it to you through the use of time-lapse films. These organisms are basically amoebae, and the amoebae are separate, and they feed on bacteria. And the interesting thing is that they do all their feeding first, so that they actually clean the bacteria off this petri dish in an area, and once they're starving, then they go on to the next stage. And in the next stage, they all stream together by this remarkable aggregation. And this is something we're going to talk about in more detail, because there's terrific cell communication at this point. And once this aggregation occurs, you get masses of non-growing cells, and these have a front end and a hind end, and they're called slugs. And after they've wandered around towards light, towards heat, they will begin to differentiate. And when they differentiate, they form only two cell types, and those two cell types are spore cells and stalk cells. And final differentiating structure is a little fruiting body that sticks up in the air, and it is a stalk, and on top of the stalk there is a minute little spore mass. Now what I would like to do is to describe two important aspects of this development, important because of the fact that they reflect fundamental development problems. Now, if you look at a slug, you see with these vital stains that there is in the anterior end a different color. In fact, it's a dark color using a red dye that doesn't exist in the posterior end. So that now you have these early signs of differentiation. We know that these red cells are going to turn into the stalk, and these cells are going to turn into spores. Now the question is everything decided, or can we change the mind of this slug? Now, long time ago it was shown that if you take one of these slugs and cut it up into pieces, each bit somehow or other magically seems to make a complete slime mold. Each little portion of that original slime mold now is a diminutive slime mold, each with its stalk and its spore mass. Now, if you think about that for a minute, that means that all the information for a slime mold is in these cells, and in these cells, and in these cells, so that stalk cells can turn back into spore cells, and spore cells can turn into stalk cells. Now, let me make an analogy. This is supposing we had a bar magnet here, and as you know, a bar magnet has a north and a south pole. Now, if we took that hacksaw and sawed this magnet into three pieces, what we would immediately discover, as you know, that we would have three little magnets, each one with a north and a south pole. And this is a very good analogy because of the fact that it shows that each portion of this slug is polarized. Each portion has all the information, all the genetic information it can make both stalk cells and spores. Now I'd like to turn to our second major problem of development that I want to talk about, and that is the problem of cell communication, cell signaling. How is it that cells can signal to one another, and what is achieved by this? Fundamental and important problem in development that many, many people are working on. Now, it just so happens that the slime molds illustrate this in the aggregation stage. In the aggregation stage, you have a whole series of cells that somehow communicate with one another so that they all come into one aggregated central cell mass. How do they do this? In the first place, it's known that there is a key chemical substance, and this key chemical substance is the attractant. This substance is capable of orienting the cells, of attracting them, so that it steers the cell into central collection points. How does a chemical gradient orient a cell? The answer is that there has to be more molecules of the chemical hitting this end, and there are receptors which receive the molecule all over, but there have to be more hitting at this end than there are at this end or anywhere else. And when that happens, then the cell apparently knows which direction to go in. Let me now draw a lot of amoebae which are sitting in a plate. And now we'll put a large gradient over this. Then all these cells on both sides move into the center and pile up. How does it happen in the beginning? Because in the beginning one has a group of cells which are separate from one another. What can happen is that this cell can suddenly give off a puff of cyclic AMP. When that puff occurs, the cell right behind it now receives molecules on its anterior end. So it has the signal to move in that direction. But soon, though, this gradient becomes obliterated. So it's very important that that cell go into a refractory period, and it only can receive the signal in the beginning, and then it ignores all further signals. But there's one other remarkable thing, and that is that once this happens to a cell, it makes its own puff of cyclic AMP, and now this new puff of cyclic AMP stimulates the next cell to move in this direction. This is precisely what happens. These pulses are waves of forward movement which move from the center outward. And in this way, you can get large numbers of cells spread over a great distance, all to go to one central point. It is particularly exciting when you realize that this is just the kind of system that we believe exists in many higher organisms, including ourselves. But one must be very careful, though, about translating information from lower forms, from simple organisms to higher organisms. And I think perhaps it's much better to say that these model systems tell you what questions to ask in the higher systems. And if one puts it this way, then I think one can say that the slime molds so far have really been very successful in this regard.